Hi guys, am I audible, visible? Hello. Am I audible and visible? Am I audible and? Hello, happy morning. Hi everyone. Happy morning. Welcome to Unacademy NEET English. First of all, let me know, am I perfectly audible and visible to you all? Yes? Am I audible and visible to you all? Okay, so welcome to Unacademy NEET English. I'm your biology educator Ambika Sharma, and today, yes, we are going to continue the principles of inheritance and variation. I know you must be thinking that, ma'am, it is the part 5th, right? 4th or 5th part. Then when are we going to finish this chapter? But we will finish it tomorrow. Tomorrow, in next class, we will only discuss about the disorders. Okay, we'll start with the pedigree analysis and then we will talk about the disorder and the chapter will be over. Okay, and the chapter will be over, and then after that I will ask you people, you please tell me when do you want the one shot of principles of inheritance and variation, because this is what I believe that no doubt this chapter, we have completed this chapter in many parts, right? We have covered the major portion of this part. We are going to cover each and everything from this chapter, but when you will revise it all together, that is going to help you out. Okay, and along with that we are going to solve the questions as well. What say? In that one shot I will include the questions as well and we will revise accordingly. Fine, so do let me know about that. And I think the next chapter should be molecular basis of inheritance. What do you people say? According to me, the next chapter should be molecular basis of inheritance, right? Wow, dekha. Navansh, this is what we were thinking. So next chapter is molecular basis of inheritance. So hopefully next week or on Saturday, we'll be starting molecular basis of inheritance. Just stay tuned to our channel, right? Subscribe our channel and you people will get to know about the schedule. Fine? And happy Ganesh Chaturthi to all of you. Okay? Which portion, bache Ridigna? Which portion? Or here you guys can see, wow. Thank you so much, Sahiti. Thank you. Right. Thank you so much for this homework, bache. Very good. So, Nandini, excellent. Right. You explained the numerical as well. Very good. I'm proud of you. Examples of multiple alleles and the genotypic ratio of the trihybrid cross as well. So, in the last class, we were talking about the polygenic inheritance, your trihybrid cross in skin color, and you know, right, that 7 different phenotypes are there. But when it comes to the ratio, okay, that proper phenotypic ratio, so you know how are we going to calculate it. Fine? Fine? Yes. So, thanks Vidisha, thank Akansha, Madhu, Priyanka, excellent. And I want to see more names here in the comment section, but it will hardly take your few seconds. Please make it a habit. If you're liking a session, please put that in the comment section. Okay? Please put that in the comment section. Yeah, I will go for the one shot as well. Don't worry about that. So now let's start the chapter and let's start the chapter. So let's discuss the schedule first. That what are we planning for this particular week? So yesterday I was not able to take your class because of some emergency, but today we will continue. Okay, so the first thing is, see, tomorrow we'll finish this chapter, principles of inheritance. So Thursday, if the slot will be available in the morning, Thursday we will start molecular basis, and here we'll start the basics of the DNA. Okay, this is what we are going to start first. So in molecular basis of inheritance, the first thing that you need to understand is basics. What is a nitrogenous base, right? What is the purine? What is the pyrimidine? Points like that you need to understand. That's the first thing. Okay. So Thursday, you know that we used to have one session. We used to have one session at 3 p.m. also, right? So might be I will take it in the morning only, and yeah, so I will take it in the morning only, and we will start this particular chapter. And when it comes to the Friday, right, it is our marathon day. Remember, it's our marathon day. So the next chapter that we need to start for marathon is body, fluid and circulation from your class 11. So this week, let's finish principles of inheritance and variation. Let's start molecular basis of inheritance, and let's start the one shot of, let's complete the body, fluid and circulation. Fine? Okay? Done? So let's go. So today, what are we going to start? We are going to start with the chromosomal theory of inheritance. That is our topic for today. Rest everything we have completed, and I'm assuming that you guys are practicing the questions as well. Fine. So let's go and let's start the, yes, I have already shared the PDF, guys. So let's start this part, that is your chromosomal theory of inheritance. So tell me, what do you know about this theory? Yes, so today you need to know about the meiosis as well. Then you will understand this topic in a wonderful way. So what are we going to start? We are going to start the chromosomal theory of inheritance, and this chromosomal theory of inheritance was given by Sutton and Vowry. They worked independently, right? They worked independently, and you know, their findings were okay, their findings were same. So this is what we are going to discuss today, the chromosomal theory of inheritance. So proposed independently by Sutton and Vowry. Is that clear? Is that clear? So this information written here is also very important. Okay, see, Mendel published his work on inheritance of characters in 1865. You know about his work, right? For 7 years, he worked on the hybridization of pea plants. Then he was able to give us that basic laws of inheritance, right? You know about it, right, guys? Guys, your energy is very low in the chat section. I know when we have so many students, like 100 plus, 200 plus, then we used to see the chat section, you know, where that enthusiasm is there, where people even talk about the doubts and everything. But it's okay, it's fine. If less number of students are there, I know if you are giving me your time in the morning, right, when you can prefer, you can just go and sleep. When you can prefer your sleep, then this chapter, if you are still here, means you want to learn something. So I want to see that excitement in the chat section. I want to see that energy in the chat section. If I'm asking one question, I want you people to answer that, and you please ask questions from me. Okay, so it should be a two-way interaction. What say, guys? It should be a two-way interaction, isn't it? Isn't it? So now you know that Mendel worked on pea plant for 7 years, but still he didn't get the recognition. So whenever you feel demotivated, just think about Mendel. Whenever you feel that, oh my God, I'm studying so hard, but I'm not able to remember the examples of animal kingdom, what to do in physics numerical, then remember one thing, remember about the Mendel. Okay. And second important thing that you all can keep it in your mind, that plants, they used to put a lot of efforts to absorb the water, okay, from the ground, right? Then they, against the gravity, they will move it up, and they can only use 1% of that water, or even less than 1% of that water. Okay, right? So they are putting a lot of things, a lot of efforts there, and they can just use 1% of that. Okay, are you getting it? So just think about it and just focus on one thing, that is efforts in a proper direction. Fine. So Mendel get recognition after his death, and when, see, it remained unrecognized till 1900, and you know, three scientists, Hugo de Vries, Karl Correns and Erich von Schemack. Okay, Erich von Schemack, they rediscovered the Mendel's work, isn't it? They rediscovered the Mendel's work. Clear, bache? Clear, bache? So they rediscovered Mendel's work. Parallelly at that point of time, work was going on the chromosome, right? Scientists, they observed that chromosomes under the microscope as well. So they tried to, you know, many people, they studied it, right? They came up with the result. Obviously, we only know about the successful results, isn't it? Isn't it? So accordingly, yes or no? Accordingly, what we have seen, right? We have seen that during this time period, right, scientists, they were also studying the chromosome, the genes pattern as well, right? So they observed that even that chromosomes, right, even that chromosomes, there is a parallelism in between the behavior of chromosome and in between the genes, in between the inherited traits. Are you getting it? That's what they observed. So see, firstly, at that time I told you communication was not easy. This is what we have already covered and already discussed, right, bache? Right, that why Mendel didn't get recognition for his work. Okay, okay. So now just look at it. In 1900, 3 scientists, they independently rediscovered Mendel's result, right? And as I said, by this time, due to advancement in microscopy that were taking place, scientists were able to carefully observe cell division. So this led to the discovery of structure in nucleus that appeared to be double and divide just before each cell division. A very important point. Right? So when these scientists, they rediscovered Mendel's work, microscopy was also advanced. Scientists, they were studying the chromosome behavior under the microscope. This is what they have observed, that just before the cell division, these chromosomes, they divide. Okay, okay. So why the word chromosome is used? Chroma means color. So they are the colored bodies, right? Right? They are the colored bodies. Are you getting it? Right? You can only see them when you can visualize it, right? You can only visualize it because of that color, because of that stain. They pick up the stain. Okay, they pick up the stain. Now you have one homework, right? And I want to see it in comment section. The homework is, you have to tell me the name of the stain that is going to color your chromosome, right? Name that stain which is going to color your chromosome. Clear, bache? Yes, bache, Aliza, right? These notes are more than enough for you. Okay, right, bache? So by 1902, the chromosome movement during meiosis had been worked out, and your Walter Sutton and Theodore Wovery, they noted that the behavior of chromosome was parallel to the behavior of genes and used chromosome movement to explain Mendel's law. So this part you need to highlight, this part, right? In 1902, what happened? That Wovery and Sutton, and remember, they worked independently, right? They were not working together, they worked independently, and this is what they studied. Fine. So they studied the chromosome behavior, and with that chromosome behavior, they tried to explain what? They tried to explain the behavior of the genes. They tried to explain the Mendel's laws. Clear, bache? Clear, bache? So that's important. Fine, that's important. MCQ can come from this part. Fine. Okay, so now let's quickly revise the division, cell division. Fine, then you guys can also understand it. You know that we are diploid organisms, isn't it? What are we? If I talk about the humans, in our case, we have 46 chromosomes, right? So we have chromosomes and they are in pair. 23 chromosomes are from mother and 23 chromosomes are from father, isn't it? 23 from mother and 23 from father. That's what we have. We are diploid. And you know that during gamete formation, okay, so 46 chromosomes are in diploid cell, isn't it? It's our 2n condition. 2n is equals to 46, so n, that's why it is 23, isn't it? N, that's why it is 23, Nandini. So during the gamete formation, so you know that for gamete formation, which division is there? It is your meiosis. What is there, Sahiti? It is your meiosis. And we know that meiosis is the reductional division. Fine. It is the reductional division. It used to take place in 2 rounds. Remember this part, it takes place in 2 rounds. It is the reductional division. So what is happening basically? Your 46 chromosomes. After meiosis 1, you will get 2 haploid cells having 23 chromosomes, and after meiosis 2, you will get 4 haploid cells, right? You will get 4 haploid cells. Clear? Clear? So what you people can observe here, you guys can see that during gamete formation, right, when there is the meiosis, during gamete formation, what is happening? From this pair of chromosome, right, these chromosomes are getting separated. Isn't it, Adiba? They are getting separated. Isn't it? As I said, pairs are there. So let me explain this. So you know that homologous pair of chromosome is present, yes or no? And what is the meaning of homologous pair of chromosome? Right? They are the chromosome having same size, same shape, same centromere position, same genes, but alleles can vary. Right? When we say homologous pair of chromosome, same, homo means same. So these are the chromosomes having same centromere position, same shape, same size and same genes. Okay, so you know that, let's say, if it is a chromosome, if it is a homologous pair of chromosome, so here a gene A is present, and you know that the position of gene on a chromosome, we call it as locus. What is it? It is locus. Okay, and the plural is loci. Clear? So if it is a homologous pair of chromosome, here if at this locus you have gene A, so here in another pair, also in another chromosome also, here you will get gene A only. So it can be capital A, it can be small a. This allele can vary, but ultimately gene is going to be same. Fine, gene is going to be same. So when there will be the meiosis and there is the reduction of the chromosome number, fine, when there will be the meiosis and there will be the reduction of the chromosome number, so one gamete will receive one chromosome out of it. Okay, one gamete is going to receive one chromosome and another gamete will receive another chromosome. Like from this pair, one gamete will receive one chromosome, another gamete will receive another chromosome. Okay, and the same way even your factors will get separated, right? When I was explaining you law of purity of gametes, okay, when I was explaining you the law of purity of gametes, that is your law of segregation, at that point of time also we discussed this, isn't it? At that point of time also we discussed it, right? That at the time of gamete formation, right, these genes, they are forming a pair, right? Genes are the pair of alleles. So we were explaining, no, that each gamete will receive only one allele for a particular character. Gametes are pure, they just have one allele for a particular character. Same behavior, right? Same behavior was observed in the chromosomes as well. So you know that this part, this chromosome will have A, this chromosome will have small a. Okay, so one gamete will receive capital A allele, another gamete will receive small a allele. So that's what they observed, right? So that's why they put forward the theory of chromosomal theory of inheritance. Clear, bache? Right? Chromosomal theory of inheritance. Is that clear? Yes. So now, okay. So now one more example, okay, to revise it. Let's say you have a cell, fine. Let's say you have a cell and this cell is having 4 chromosomes. How many chromosomes are there in this cell? Let's say this cell is having 4 chromosomes. Fine. 4 chromosomes means obviously these chromosomes are having the genes. Genes are present on these chromosomes, isn't it? Genes are present on these chromosomes. This is a cell. This is a diploid cell having 4 chromosomes. So now you know that, right? Right. Obviously at that point of time when Sutton and Bovary, when they studied it, right, they were not clear with all the facts. But now you know more things, isn't it? Isn't it? So I'm using that information and accordingly I'm explaining this concept. Fine. Accordingly I'm explaining this concept. So you know that a cell before division, right, there first of all there will be the interphase, right? There is the interphase, the resting phase where, you know, cell is preparing itself for the division. Okay, right now no doubt I'm showing the chromosomes like this, but you know when cell is in the interphase, okay, you know when cell is in the interphase, this genetic material, it is bache tangled. This genetic material is not condensed at all. You know about it, right? You know about it. Yes or no? No doubt I'm giving you the example of a cell where the number of chromosomes are 4, but you know when it is the resting state, when it is the interphase, right? Recall the meiosis, recall the cell division now. So in interphase you have G1 phase, S phase and G2 phase. Remember? G1 phase, Active protein synthesis, RNA synthesis will be there. S phase, the only phase where your DNA replication will be there. Then G2 phase, again protein synthesis, RNA synthesis will be there. So when a cell is in the interphase, you know that these chromosomes, they are not condensed at all, right? They are not condensed at all. Fine. They are present like threads. They are tangled. And slowly after G2 phase, when it will enter in M phase, then it is going to arrange itself, isn't it? But one thing that we know, that in S phase, there is the DNA duplication, there is the replication of the DNA, and your monad chromosomes, they will become dyad, right? Remember this part. Your monad chromosome, they will become dyad. So if I am showing chromosomes like this, right, don't be like, ma'am, it is a chromatid. No. It is also a chromosome, but it is in its monad state when there is no DNA duplication. Okay, when there is no DNA duplication, fine. When there is no DNA duplication, it is that state. Okay, okay. After S phase, it will be in this phase. You know it very well. So if I am taking the example of 4 chromosome, so after S phase, I will take it now, right? So obviously they are in it, they are dyad state. They are like this. They are like this, right? So in prophase, they are going to behave like this, yes or no? Right? In the prophase, your chromosomes will become condensed. So they are going to behave like that. Okay, so now you know, here we are talking about the gamete formation. So we will be discussing meiosis, and meiosis 1 will be there initially. So meiosis 1 is having prophase 1, it is having metaphase 1, it is having anaphase 1, it is having telophase 1. And after the interkinesis, there will be meiosis 2 having prophase 2, having metaphase 2, having anaphase 2, and finally the telophase 2. So that's why you are getting 4 daughter cells, because in meiosis there will be 2 rounds of nuclear division, 2 rounds of cytoplasmic division, but there is only 1 round of DNA replication. Clear? 1 round of DNA replication will be there. So meiosis 1, meiosis 2 is there. So prophase 1, metaphase 1, anaphase 1, telophase 1. Prophase 2, metaphase 2, anaphase 2, telophase 2 will be there. So prophase 1, you know what do we have here? It's LZPDD. Right? It is having LZPDD. Leptotene, zygotene, pachytene, diplotene, dikinesis. That's what we have, Trisha, here. That's what we have here. Right? We have 5 substages in the prophase 1. So ultimately what I want to say here, right? If I not, see, we have already covered cell cycle and cell division. I'm assuming you know that much. But this is just for your quick revision. Why? So that you can understand this topic in a better way. Fine, bache? So here, what am I saying? All I'm saying is, when there is the gamete formation, you know that meiosis 1 and 2 will be there. And in prophase 1, we have the substage, that is your zygotene, where there will be the pairing of homologous pair of chromosome. Isn't it? Where there is the pairing of homologous pair of chromosome. So if a cell is having 4 chromosome, right? So you have 2 pairs of homologous chromosome. Let's take it like this. Okay, that you are having 2 pairs of homologous chromosome, right? So in the prophase 1, basically condensation will be there, right? Condensation will start, pairing of homologous chromosome will be there, that crossing over and everything will be there, you know that, right? But Sutton and Bovary, they just studied the behavior. Okay, okay, and you know that after this, when you talk about the anaphase 1, right? I hope you can relate it. Right, metaphase 1, you know what is going to happen in metaphase 1. In metaphase 1, these pairs, right, in metaphase 1, the pair of chromosome, you know that it will arrange itself like this. It will arrange itself on the equator, at the equator, right? Right, so metaphase means the chromosome, they will come here in the center. They are going to arrange themselves at that equator, right? So 2 metaphasic plates will be there in the metaphase 1. 2 metaphasic plates will be there in the metaphase 1, because these homologous pair of chromosomes, they are attached together, they are paired, right? So they will arrange themselves on these metaphasic plates at the equator. Now when it is the anaphase 1, of course after anaphase 1, in anaphase there will be the separation, right? There will be the separation after anaphase 1. So after anaphase 1, if you remember, what will happen in anaphase 1? What is going to happen in anaphase 1? It's not anaphase 2, it's anaphase 1. So what will happen here in anaphase 1? Anyone in the class? Right, there is the separation of, right, there is the separation of this pair of chromosome. This bivalent pair, it will get separated from each other. So after anaphase 1 or during anaphase, actually this is the scenario, bache. Right, this is the scenario when you are talking about what? When you are talking about the anaphase 1. So from this, so this pair of homologous chromosome will get separated from each other. Fine, this pair of homologous chromosome, it will get separated from each other. And after telophase 1, right, after telophase 1, during telophase, you know that it is generally the opposite of prophase, right? So after telophase 1, right, after that cytokinesis, you will see this condition, right? You are going to see this condition, right? For your better understanding, let me change the color here. Okay, let me change the color here. So basically when you talk about the gametes, in the gametes, that is what you will see, bache. That is what you will see, bache, right? So you will see, yes, you will see that each gamete will receive, from that pair, each gamete is going to receive only one chromosome. Okay, okay, they were in pair, so the pair is getting separated. Fine, it is the pair which is getting separated. Now clear? Yes, now clear? Right, so now let's move to the, so now let's study one more thing here. So see, these are the chromosome, one pair, and this is the second pair. So now just look at it. Here, bache, if there is a, here if let's say small is there, here let's say capital C is there, here let's say small c is there. This is how the genes are going to present, right? That's how the genes are going to present. Yes or no? Like if this chromosome, it is having a gene, right? If it is having a gene, okay, because this is the homologous pair of chromosome, the another chromosome is also having the a gene. The allele can vary, but the gene is going to be same. Here if it is the B, here also it is going to be B. The allele can vary. So when there is the meiosis 1, after meiosis 1, after cytokinesis, see, this one gamete is having capital A, another is having capital A, this one gamete is having capital B, another one is having small b. So that's why we used to say your gametes are pure. Okay, they have just one allele. They have just one allele for a particular character. Fine, they have just one allele for a particular character. So after, yes students, after your anaphase 2, what will happen after anaphase 2 and telophase 2? You will get 4 daughter cells, you will get 4 haploid cells, then ultimately it is related to the C. So here ultimately we just need to divide the DNA content equally, that's all. That's all. Chromosome number will be same, but this is what you are going to do. So see, this particular gamete will have capital A, this one is having capital A, this one is having small a, this one is having small a. Here capital C, small c, small c. If here it is having capital B, small b, small b. So can you see this? Can you see this? So chromosome and that genes, they are behaving in a same manner. Like we are saying that at gamete, at the time of gamete formation, factors will get separated. See, even the chromosomes are getting separated. Capital A, small a. Capital C, small c. Is that clear or not? Right? Gokul bache, we'll post the plant kingdom part 2. Kindly do not spam. Done? So now compare it here. See, look at this diagram. So they are showing meiosis and germ cell formation in a cell with 4 chromosomes. Okay, can you see these 4 chromosomes? 1, 2, 3 and 4. Okay, 1, 2, 3 and 4. Done? Done? So after G1, S, then G2. S will be the phase where synthesis will occur. So after synthesis, this is the condition that you are going to get. Okay, okay, so you guys can see that this is the homologous pair of chromosome. Fine. Fine, bache. So in prophase 1, in zygote, synapses will occur, and you know that in meiosis 1, anaphase, right. So this homologous pair of chromosome will get separated from each other. It will get separated from each other. This one here, this one here, this one here, this one here. Fine. And when it is the anaphase 2, okay, when it is the anaphase 2, further there is the splitting of centromere, there is a separation of chromatids, so accordingly it is taking place. So germ cells, they are the haploid cells, right? So initially 2 chromosomes were there, now 4 chromosomes were there, now 2. Each gamete is having 2 chromosomes. Is that clear? Is that clear? That's how it is getting separated, right? So same thing I have explained here. So see, initially A, okay, let's say here you have small a, here you have capital B, here you have small b. So this one together, both these sister chromatids, capital A, small a, capital B, small b. Now it is getting separated, right? So this cell will have capital A and capital B, this cell will have small a, small b. After anaphase 2, this cell is going to have, yes, right, see, after anaphase 2, capital A, capital A, capital B, small a, small b, small a, small b. That will be the scenario. Fine, that will be the scenario. Okay, okay, so this table is given here in your book, right? See, they are saying, so comparison table is given. They occur in pairs, they occur in pairs. Both gene and chromosomes, they occur in pairs. First thing. Secondly, they segregate at the time of gamete formation such that one of each pair is transmitted to a gamete. Here, segregated gamete formation and only one of each pair is transmitted to a gamete. See, both behavior is same. Next, independent pairs segregate independently of each other. Like let's say if you are talking about 2 different genes, capital A, capital B, obviously they segregate independently of each other. They are not affecting each other's segregation. So from this point, I can conclude that here they are talking about the gene and this B is for the chromosomes then. This is a comparison table, na? This is a comparison table, isn't it? Isn't it? See what they mentioned, independent pairs, right? Independent pairs means this, right? They segregate independent of each other. The segregation of A is not affecting the segregation of B. And here they are saying one pair segregates independently of another pair, right? So they are talking about the homologous pair, that one pair of homologous chromosome is segregating independently from the another pair of the homologous chromosomes like this. So A is the gene here, B is the chromosomes here, right? So this is for the genes, this is for the chromosomes. It is important. Fine, bache, it is important. Clear? Okay. So see, this is something from my side for your quick revision. So see, chromosomal theory of inheritance, Sutton and Boveri. So like the hereditary traits, chromosomes, right, they retain their number, structure and individuality throughout the life of an organism from generation to generation, isn't it? From generation to generation, isn't it? Isn't it? Yes or no? Like the hereditary traits, the chromosomes, they retain their number, structure and individuality throughout the life of an organism from generation to generation. It's a fact. So two neither get lost nor mixed up. They behave as units. They behave as units, right? That's how you can compare your genes and the chromosome. Next, both chromosomes as well as genes, they occur in pairs in diploid cell. The 2 alleles of a gene pair are located on homologous sites on homologous chromosomes. Understood? Understood? When you are talking about the genes, when it is a homologous chromosome, so they are present on the right. They are occurring in pairs, so they are present on homologous sites. Next, a gamete contains only 1 chromosome of a type and only one of the 2 alleles of a trait. This comparison is good, Trisha. This comparison is good, isn't it? One gamete contains only 1 chromosome of a type. Of a particular type, only 1 chromosome of a particular type will be there and only 1 allele from that pair of alleles. Clear? Clear? Okay, so the condition of both chromosome as well as Mendelian factor is restored during fertilization. Fine, bache? Fine. So the paired conditions, right? The paired condition of both chromosome as well as Mendelian factor is restored during fertilization. So these are the points, right? They are very good for recalling chromosomal theory of inheritance. Done, bache? They are very important for recalling that. So now we will discuss the experimental verification of this theory, which was given by your father of experimental genetics. Can you tell me the name of that person? Who's the father of experimental genetics? Who's the father of experimental genetics? Exactly. Bache, father of experimental genetics. Hishanvi. Bache, father of experimental genetics, T.H. Morgan, Thomas Hunt Morgan. Okay, so Thomas Hunt Morgan gave the experimental proof for the chromosomal theory of inheritance. He worked on Drosophila melanogaster, which is your fruit fly, and Morgan has given the chromosomal theory of linkage. So do not confuse it. If it is chromosomal theory of inheritance, Sutton and Vovry. If it is chromosomal theory of linkage, then it is T.H. Morgan. Okay, then it is T.H. Morgan. Fine. So, bache, can we revise this part from NCERT? Yes. NCERT reading is also important, no? So can we just revise it from NCERT? Okay, so see this. So recall that you have studied the behavior of chromosome during mitosis and meiosis. The important thing to remember are that chromosome as well as genes, they are in pairs. Fine. So see, so during anaphase of meiosis 1, already discussed, isn't it? During anaphase of meiosis 1, two chromosome pair, they can align at the metaphase plate independently of each other. Obviously, these chromosomes, they are arranging themselves on that at that metaphasic plate independently of each other. Fine. So compare the chromosome of 4 different color in the left and right column. It is as per the diagram. So left column, possibility 1, right, where one long orange and short green chromosome and long yellow and short red chromosomes at the same pole, and possibility 2, one long orange and short red chromosome and long yellow and short green chromosome at the same pole. So basically they are talking about this. Okay, done, bache? So this is what they are explaining, right? And next topic is your chromosomal theory of linkage. This is what we need to understand. Fine. So Sutton and Vovry argued that pairing and separation would lead to the segregation of pair of factors they carried. So Sutton united the knowledge of chromosomal segregation with Mendelian principle and called it the chromosomal theory of inheritance. Okay, so see how things are related, right? Mendel worked on such things many years before, right? They studied it, they compared it with their findings, and this is the conclusion that they do. Okay, so see the beauty of science here. So now, following this synthesis of ideas, experimental verification of chromosomal theory, it was given by Thomas Hunt Morgan, another important MCQ, right? His colleagues and he led to the discovering the basis for the variation that sexual reproduction produced. Clear? He led to discovering the basis for the variation that sexual reproduction produced. This line is very important, bache. Okay, in statement based question they can ask you such points. Fine, they can ask you such points. Clear? So Morgan worked with the tiny fruit flies Drosophila melanogaster. So basically Drosophila is the first animal where Mendel's laws were applied. Okay, Mendel studied the plants. Okay, and Thomas Hunt Morgan, he applied that laws on the animal, and Drosophila is the first animal there. Drosophila melanogaster, which is fruit fly, near about 2 millimeter. And you know this fruit fly, it like whenever there is a fruit, the ripe fruit, your mango is there. So if you will put the yeast extract on that mango, Drosophila will come immediately. It will get attracted towards it. It will come immediately. So they used Drosophila for that. Okay. Okay. So Drosophila is very much suitable for such studies. Do you know why? Do you know why? Because of its size also. Because it is very easy to rear and bred in the lab conditions. Fine. And when you look at the Drosophila, right, male and female, you can differentiate them very clearly. Right? You can differentiate them very clearly. When you talk about the female, female is large. Female is having ovipositor as well. Okay. Okay. So you can even differentiate male and female by just looking at them. The sexual dimorphism is there. These conditions are important. The sexual dimorphism is there. And the very important point to deal with the Drosophila is it's very easy to handle. Right? Easily under the lab condition, it can be bred, okay, and reared. Fine. On synthetic medium, you can grow it. You can grow. And moreover, they are having contrasting pair of characters. Very, like that characters are visible, right? Very easily you can see such characters. They have that contrasting pair of characters as well. Okay. And you know, one progeny produce thousands of, right? Like in one parents, they produce thousands of offsprings in the progeny. That is also very good. Like pea plant, we don't have to wait for a long period. Very immediately you will see. Why? Because the life cycle is also of 2 weeks, right? 2 weeks. When it comes to the life cycle of the Drosophila, it is also important. It is 2 weeks. Okay. And number of chromosomes are 4. 4 pairs were there or 4 chromosomes were there? Can you tell me? Right? Can you tell me when it comes to the Drosophila, 4 pairs of chromosomes were there or 4 chromosomes were there? Bache, 4 pairs. Okay. 4 pairs of chromosomes. So they are morphologically distinct chromosomes. They are morphologically distinct chromosomes. So they could be grown on simple synthetic medium in lab. They complete their life cycle in about 2 weeks. Single mating can produce large number of progeny flies, and clear differentiation of the sexes, the male and female, means sexual dimorphism is there. Okay. And it has many type of hereditary variations that can be seen with the low power microscope. So as I said, that contrasting alleles, they are very easily to notice. Clear? They were very easy to notice. Okay. Okay. Right. And moreover, this is extra. You know, in the case of Drosophila, you also see polytene chromosomes. What type of chromosomes you are going to see in the salivary glands? Polytene chromosomes. You know, large chromosome that chromonemata is there, right? I will not teach you this part in detail about the polytene chromosomes. Again, that's your homework. You will write down the definition of polytene chromosomes in the comment section. Okay. In the comment section, you will tell me about the polytene chromosomes. So they are poly means many, right? You can say that that chromatin material is too much. That's how you can relate. So these polytene chromosomes were very good to, you know, to see the mutations. Okay. Okay. So that is also an advantage. And even in Drosophila, XX and XY type of sex determination is there. And you know, it is just like the humans, XY male, XX female. So that thing is also there in the Drosophila. Okay. Okay. So heteromorphic sex chromosome in males, it was very important to, you know, it was helping to understand the chromosomal behavior more in a better way. All right. So polytene chromosomes were there in the salivary glands of larva bache, right? So they helped to understand the mutations, the chromosomal aberrations. Fine. So that's why they use the Drosophila. So Drosophila is the first animal where, you know, the Mendel's laws were applied. And you know what Morgan observed? Do you know what Morgan observed? So as per Morgan, right, when he applied laws on Drosophila, so he was not getting he didn't get the ratio same like Mendel. He didn't get the ratio same like Mendel. Like, you know that when it is the dihybrid cross, this is the ratio of dihybrid cross. As for Mendel, this is the ratio of dihybrid cross, but he didn't get the ratios like this. Instead of that, even in dihybrid cross, he noticed that the ratio is just like monohybrid cross, 1 point. And moreover, he saw that, as Mendel was saying, right, that chromosomes, two different genes, right, two different genes, they do not affect each other's segregation. They segregate randomly. But he didn't observe such things, right? He observed that genes, they are staying together from generation to generation. And then, bache, he explained what? He explained the linkage, the phenomena of linkage. Is that clear? He explained the phenomena of linkage. So linkage is tendency of some of the genes to inherit all together. What is linkage, bache? It is the tendency of the genes to inherit together. Is that clear? Is that clear? But you know, Morgan was not the first one to study it and to, you know, explain it. He, no doubt, he gave the chromosomal theory of linkage. He explained it, but he was not the first one to observe it. Do you know that? He was not the first one to observe it. Actually, the principle of linkage, you know, the principle of linkage, it was first discovered by Watson and Punnett. You know, they studied it first. Do you know that? They studied it first. Watson and Punnett, they studied it first. Yes. They were working on sweet pea. Fine. That is your Lathyrus. They were working on your Lathyrus. Okay, Lathyrus pieces. And they observed, right, when they studied the dihybrid cross, they didn't get this ratio. Instead of that, they get the ratio 7:1:1:7. That's what they got. You know, that's what they got. Right? Okay. So dihybrid ratio was different. And even when I was teaching you test cross, I told you that, right, that the ratio of test cross, right, in a dihybrid, in the 2-gene inheritance is this. In 2-gene inheritance, this is the ratio of the test cross, but they didn't get it. They get this ratio. Okay, and what were they studying? See, this one. As I said, Lathyrus. So these characters were taken: blue flower, long pollen; red flower, round. Right? Blue flower, long pollen; red flower, round pollen. So just look at it. So this is the ratio. Expected ratio is this, but observed ratio is this. Okay. So parent-like traits are more. See, blue long, red round. Parent-like characters were more, and that recombination or recombinant, they were not that much. Fine. So that's why this is the ratio. Got it? That's why this is the ratio. Done. So in order to explain it, right, in order to explain it, Watson and Punnett, they put forward the theory of coupling and repulsion. Right? They put forward the theory of coupling and repulsion in this case. You know, this is what we study in linkage. We talk about the coupling and repulsion in linkage. Do you know about it? Yes. Do you know about it, coupling and repulsion? So see, when it is the coupling, what were they saying? Like, let's say when this is the scenario. Just understand it. When this is the scenario, when it is coupling, so they were saying then that when A and B, they are coming from the same parent. What this coupling is explaining, it is saying that capital A and capital B. See, capital A and B, when it is coming from same parent, so they will stay together in the next generation as well. As per this coupling, according to this theory, when two genes, for an example, A and B, right, they come from the same parent, they tend to enter to the same gamete. Okay, they tend to enter in the same gamete and they will inherit together. Okay, these two genes, they will inherit together. Is that clear? So they come from same parent, they tend to enter the same gamete, so they will be right? They will be inherited together. Are you getting it? They will be inherited together. Is that clear? Okay. And see, when it was repulsion, so here they were saying that when this is the scenario, A and B, they are coming from different parents, right? When A and B, they are coming from different parents, they tend to enter different gametes. So this is what repulsion is. So that's how Watson and Punnett, they tried to explain it. Okay, that's how this Watson and Punnett, they tried to explain it. So first of all, tell me this part clear? Yes, this part clear? Sure? Tell me, right? So after all this, bache, your Morgan carried out the experiments. Okay. Okay, so after all this, your Morgan carried out several dihybrid crosses in Drosophila to study genes that were sex-linked. The crosses were similar to the dihybrid cross carried out by Mendel in peas, right, bache? But the result he obtained, they were different. Fine, bache? Clear? Yes, is it clear? Sure. So now this is the diagram that we need to understand, right? This is the part that we are going to discuss now. So up to this part, if there is any doubt, do let me know. Up to this part, if there is any doubt, do let me know. And what is the definition of the linkage? It is the, yes, what exactly is the linkage? Tell me, the tendency of some of the genes to inherit together as a unit. What is linkage? The tendency of some of the genes to inherit together. Clear, bache? To inherit together is known as your linkage. Fine, it is known as your linkage. Okay, okay. So now before explaining this part, let me tell you. See, now if linkage is there, then no independent assortment. I told you now that law of dominance and law of independent assortment, it is not universally accepted. It is not universally accepted. Okay, law of segregation, law of purity of gamete is universally accepted. Otherwise, these two laws, they are not universally accepted. So I am saying that when there is the linkage, there is no independent assortment. Actually, what we have studied in independent assortment. In independent assortment, that's what we used to say na bache, that if two genes are there, right, they are not affecting each other's segregation at the time of gamete formation. This is what we used to say, that at the time of gamete formation, the segregation of A is not affecting the segregation of B. Randomly they are segregating. Independently, right? Independently they are segregating and randomly they are meeting in the next generation. When there is the formation of diploid, randomly they are meeting and, right, independently they are segregating from each other. Okay, but what we see in linkage, we say that two genes, they stay together. Right, two genes, they stay together from generation to generation. So obviously if linkage is there, then there is no independent assortment. And if there is independent assortment, means linkage is not there. Means linkage is not there, that's the point. So what is the thing that you need to understand here, bache? That independent assortment is possible when two genes, they are present on different chromosome. Okay, they are present on different chromosomes. They are not present on same chromosomes. Are you getting it? They are present on different chromosomes. They are not present on same chromosome. Then there is the independent assortment. And moreover, even if they are present in the same chromosome, there should be distance between these two genes. Okay, okay, so that crossing over can occur. Fine, so that crossing over can occur. So what is crossing over? It is the recombination. It is the recombination. You know that during packaging, right, there is the exchange of segments of, right, non-sister chromatids of homologous pair of chromosomes. Isn't it, Nandini? During the Trisha, during the gamete formation, during your meiosis, during your packeting, what is going to happen? There is the exchange of segments of the non-sister chromatids of the homologous pair of chromosomes. Isn't it? Of the homologous pair of chromosomes. Isn't it? Isn't it? Right. So recombination is only possible when there is a distance between the genes if they are located on the same chromosome. So that's what you need to understand. So these are some common points that you should know about this linkage. Fine? Fine? Okay? Okay. So can I say that if two genes, they are very close to each other, they will stay together? Can I say so? If two genes, they are very close to each other, yes, there are chances, right? They are very close to each other. They are present on the same, chromosome. Then there are chances that they will inherit together. Of course, yes. So linkage is directly, okay, sorry. Linkage is what? Linkage is what? Linkage is directly proportional to the distance. Like more distance, sorry, linkage is inversely proportional, isn't it? Isn't it? When you are talking about the distance, okay, so what is going to happen? Linkage is inversely proportional. The distance, linkage and distance, this is inversely proportional. If distance is increasing, linkage is decreasing. If linkage is increasing, if linkage is there, it means distance is less. Okay, so in the case of linkage, you know that when it comes to the distance, they both are inversely proportional. Isn't it? It is inversely proportional. If one is increasing, another is decreasing. More distance means more crossing over, but less linkage. Okay, but less linkage. Clear? So this is what linkage is. The tendency of chromosomes, the tendency of genes that they will stay together and they will inherit together. Fine? That's what your linkage is. Clear, bache? That's what your linkage is. And yes, one more thing. See, this is your chromosome. So if I have to, let's say this is the diagram which is representing your chromosome. Now see, genes are present on these chromosomes in a linear order. That's how these chromosomes are present. See, that's how these genes are present. So genes are present on a chromosome in a linear order. Right, in a linear order. That's how they are present. So A and B very close to each other, they will inherit together because they have less distance. A and F, they are far from each other. Obviously there are chances that crossing over can occur and they can get separated. Isn't it? It's right. So these are the genes. So genes which are present on same chromosome, and right, understand it. No need to write such points, but try to understand it. So if genes, they are present on same chromosomes and they are very close to each other, we will call them as linked genes. What we call it? We will call them as linked genes. Genes, they are present in a sequential manner, in a proper order on a chromosome. Then they are making what? They are basically the linked genes. They are basically the linked genes. So if here you have one gene, right, and this this G is present on another chromosome, they are not at all linked, right? They are not at all linked. They are unlinked genes. Clear? They are unlinked genes. These two terms clear? What is the meaning of linked gene? What is the meaning of unlinked genes? Okay. Now, second point. See, now you know we have the pair of chromosomes. Okay. And this pair of chromosome is having same genes. If A is here, here it can be small a. If B is here, here it can be capital B also. So basically they have same genes. So this homologous pair of chromosome, it is making one linkage group. There is one more term that you need to understand, and that is the linkage groups, right? That is the linkage groups. So if chromosomes are present on a same chromosome, they are making, right, these chromosomes, they are present on same chromosome, they are making one linkage group, yes or no? Or let's compare it with the homologous pair of chromosome. So if it is homologous, if here it is A, here also gene A will be there, allele can vary, right? That's how it is present. So this homologous pair of chromosome is making one linkage group. It is having the same information of the gene, so it is making one linkage group, yes or no? Tell me, is it making one linkage group or not? It is having same type of genes. It is having same types of genes. right? So genes which are present on a single chromosomes, they are making the linkage group. Yes, they will inherit together as a group. Okay, they will inherit together as a group. So the genes which are present on, right? So genes which are present on one chromosome, they are making one linkage group. Okay, they are present together as a group. Fine. Yes or no? Yes or no? Right? So the group of genes present on the same chromosome, they are making one linkage group. So when you are talking about homologous pair of chromosomes, so ultimately genes are same. So homologous pair of chromosome is making one linkage group. Now let me explain this, that in the case of human females, how many chromosomes are there in totality? How many chromosomes are there in totality in the females? Of course, if we are humans, it doesn't matter whether that's a male or female, chromosome number will be 46. But if you talk about the linkage group here, so in the case of human female, 23 linkage groups are there. Right? Why 23 linkage groups? Because linkage group means the group of genes present on same chromosome. So when it is the homologous pair of chromosome, right, although they are two chromosomes, but you know that they have same gene. So they are making same linkage group. Clear? But on the other hand, if you talk about the males, right? So in the case of males, see, females are homogametic, right? Females are homogametic. You know that, na? That XX, we have two pair of X chromosomes. And when you are talking about the males, so males are heterogametic. That's XY. Their sex chromosomes are not same. Their sex chromosomes are not same. Okay? Okay, so rest 22 pairs, they are making 22 linkage groups, and here in the case of human males, you have 24 linkage groups. Okay, you have 24 linkage groups. Why? Because X and Y, they have different genes, they do not have the same genes, right? In the case of females, because our sex chromosomes, that pair is same, both are X chromosomes. Clear? So that's why they are making one linkage group. That's why in human females, only 23 linkage groups are there. But in the case of males, when you talk about their sex chromosomes, that is XY, that is heterogametic. So in their case, right, X is having different genes, Y is having different genes, Y is a Hollandric chromosome, it is having different genes. So human males, they have 24 linkage groups, right? They have 24 linkage groups. Is that clear? They have 24 linkage groups. Is that clear? So what is linkage? What is the meaning of linked genes? What is the meaning of unlinked genes? And what is the meaning of linkage group? Is that clear? Yes or no? If still there is any doubt, do let me know. Right? Sahiti, I'm repeating this. A pair of chromosome having same genes, it is making one linkage group because genes are ultimately same. That information is ultimately same. But in the case of males, because it is XY, X will have different genes. Y will have different genes. That's why 24 linkage groups are there. Okay. So next thing is complete linkage and incomplete linkage. Right? So complete linkage means, which is very rare. Do you know that? Complete linkage is very rare. Right? So complete linkage means ditto like parents. Right? All the genes, they are not at all getting separated. Like see, if in the parents it's A, B, C, in the progeny also A, B, C is there. Right? There is no change of allele. It's complete linkage. Same like parents, and you know it's very rare, and you will see the complete linkage in the case of male Drosophila. Do you know that? In the case of male Drosophila, you will see complete linkage, which is very rare, bache. Okay, so when genes located in same chromosome inherit together over 2 and more generations. What is complete linkage? So the genes which are present on same chromosome, they inherit together, right, for 2 to 3 generations. They are making what? They are making the complete linkage, right? What are they making? They are making the complete linkage, which is very rare, okay, which is very rare. So here in the progeny, you will not see recombinants. You are not going to see the recombinants. Fine, right? So because it is a complete linkage, so its ratio here will be like 3:1, like the monohybrid cross. Will give you one example. If it is complete linkage, so even if it is a dihybrid cross, even if we are studying the inheritance of two different genes, so that genes are going to stay together, right? They are going to stay together. So if they will stay together means they are together working as a unit. So ratio will not change. Ratio will not change. You will get the ratio like the, right? You will get the ratio like the, like what? Like what? You will get this ratio, right? Like the monohybrid cross. Fine. This is the ratio that you will get in F2 generation because they will move together. And if you talk about the linkage, the complete linkage, then this is the test cross ratio. So you need to remember the ratios. Okay. So if it's a complete linkage where there is no chance of any separation, generation to generation, same genes will pass all together. So even if you study the dihybrid cross in F2 generation, that's the ratio you are going to get because they will stay together only. And that will be the ratio of your test cross. Fine, that will be the ratio of your test cross. Clear? This is the complete linkage, which is very rare. You will see it in the case of male Drosophila. Okay, you are going to see it in the case of male Drosophila. Now when it is incomplete linkage, means there is some chances of crossing over is there. Okay, just say I gave you this example, no? This one, blue flower long pollen, red flower round pollen. So here you will not see the complete linkage. It is incomplete linkage because there are some recombinants as well. Okay, there are some recombinants as well. Fine, there are some recombinants as well. So that is your incomplete linkage. So here even you will see parental combination, they are more than the recombinants. Parental combination is more than the recombinant. Okay, okay, right? So in incomplete linkage, you can say there is some distance in between the genes that we are studying. Okay, so there are chances of crossing over. So that's why this linkage is incomplete. So now in your NCERT, the example that we are going to study now, it is also the example of your incomplete linkage. Let me explain this. So see, Morgan hybridized yellow-bodied, white-eyed female. Yellow-bodied. Body color is yellow. In the Drosophila, you will see many contrasting alleles, many different, contrasting traits, many different characters you are going to see. Yellow-bodied, but eyes are white, right? To brown-bodied, red-eyed males. Males are having red eyes, females are having white eyes, right? So they are intercrossed there and intercross their F progeny. So after this cross, they will get F1 generation. They do the selfing. So he observed that two genes, they did not segregate independently of each other, and F2 ratio deviated very significantly from 9:3:1, which is the expected ratio. So it was not same. So when you study this, so Morgan and his group, they knew that the genes were located on the same on the X chromosome, right? The experiment that we are discussing now, here it is related to that X chromosome, which is the 6th chromosome of the Drosophila. Okay, and they saw quickly that when two genes in a dihybrid cross were situated on the same chromosome, the proportion of parental gene combinations were much higher than the non-parental type. Obviously, it is understood. So if you are studying the inheritance of two genes present on same chromosome, remember this point, you will get more parent-like combination than the recombinants, right? Than the new combination. Why? Because linkage will be there. Either complete or incomplete, but linkage will be there. So Morgan attributed this due to the physical association or the linkage of two genes, and he coined the term linkage, right? To describe the physical association of genes on a chromosome, and recombination to describe the generation of non-parental gene combinations. Right? So recombination, new combinations will be there. Non-parental combinations will be there. So Morgan and his group also found that even when genes were grouped on same chromosomes, some genes were very tightly linked. tightly linked means they will show, right? If they are very tightly linked, no recombination at all, complete linkage. If there is some distance, incomplete linkage, but linkage will be there. Okay, so the genes which are loosely linked, so genes present on same chromosome, they are the linked genes. If they are very close to each other, closely linked. If they have distance in between two, loosely linked, right? So for example, he found that the genes white and yellow were very tightly linked and showed only 1.3% recombination, right? That the genes for white and yellow, they are so tightly associated. So you will, in the next generation, you will see more individual having white and yellow, white eyes and yellow bodied color. Clear? So they are only showing 1.3% recombination, and white and miniature wing, it showed 37.2% recombination. So they conducted one experiment, Nandini, and this is the result that they explained. Clear, bache? So his student Alfred Sturtevant, that's very important. I will discuss it separately. So Morgan's student Alfred Sturtevant used the frequency of recombination between gene pair on same chromosome as a measure of distance. So we will talk about it, but before that, let's see this. So this is cross A, yellow white, and this is the wild type. So here, see, yellow white, they are more close to each other. Now this is another cross, white miniature body, right, and white and miniature, right? So this is the wild type. So here, if you observe this, so here the genes that you are taking, they have more distance, and here they have less distance in between, isn't it? Here Y and W, they are closely associated to each other. W and M, they are loosely associated to each other. They have distance. So yes, there are chances here that they will show more recombination than this cross A. Okay, they will show more recombination than this cross A, right? So here you guys can see that in this cross A, parental type is 98.7%, recombinant type is 1.3%. Why? Because two genes are close to each other. But here, because there is a distance between these genes, so parental types are 62.8%, recombinant types are 37.2%. Now got it? So this is what you will see in the F2 generation. Fine. Navina, you need to join the group for that All India test series, and the syllabus is shared there, bache. Okay? Okay, so that's what linkage is. So this is a non-crossover gamete, means same like parents, capital A, capital B. This is a crossover gamete, right? Recombinant. One is capital A, another is small b. So any doubt here in linkage? Yes, any doubt here in linkage? Are you sure? Are you sure? So next is this chromosomal mapping. I told you na, the student of Thomas Hunt Morgan, Alfred Sturtevant. Alfred Sturtevant, he expressed it, right? So question can also come on the basis of that, but numerical problems can come. So see, mark this point, mark this paragraph in your NCERT. So what are they saying? That his student Alfred Sturtevant, he used the frequency of recombination. That what exactly is the frequency of recombination in between two genes, right? He used that frequency, right, frequency between gene pairs on same chromosome as a measure of distance between genes and mapped their, right, mapped their position on the chromosome. So basically what Sturtevant did, he checked that gene pairs, what is the recombination frequency in them, and accordingly he explained the position of genes on the chromosome. So he used that recombination frequency to map the position of genes on the chromosome. Clear, bache? To map the position of genes on the chromosome. Is that clear? Yes. So as per Morgan, percentage of recombinants in the progeny depends upon the frequency of crossing over, of course. And this is what Alfred Sturtevant expressed. Okay, clear, bache? Right. So the scientist name is also important. So just see this. So they are extensively used as a starting point in the sequencing of whole genome, and it was done in case of human genome sequencing. So see, that's what you need to remember. So the representation of figure of relative position of genes on chromosome, it is the chromosomal map, right? The representation of figure of relative position of genes, the representation of figure of relative position of genes on the chromosome is known as chromosomal map, and the process of identifying gene loci is the mapping, is that chromosomal mapping. So the frequency of crossing over or recombination between two linked genes is directly proportional to the physical distance. Like let's say here you have two genes A and B. So in between these two genes, the recombination frequency is 10%. What is the recombination frequency? It is 10%. So how will I say the distance? So I will say the distance between A and B is also 10 map units. You can also use the word 10 map units or 10 centimorgan. These are the units that we are going to use. 10 map units or 10 centimorgan. Clear? 10 map units or 10 centimorgan. Is that clear, Nandini? So recombination frequency is 10%, means two genes, they are having a distance of 10 map units or 10 centimorgans. Clear, bache? So this unit, centimorgan, that's how you are going to denote it. See, centimorgan, that's how you are going to denote it. So centimorgan you use in eukaryotes. In the case of eukaryotic genetics, now you use the word centimorgan. Like centimorgan and map unit, it is same, but in the case of eukaryotic genetics, you will use centimorgan. And in case of microbial genetics, right, in the case of microorganisms, when you are studying the same thing, then you use map units. Okay, then you specifically call it as map units. So when it is the eukaryotic genetics, centimorgan, when it is the microbial genetics, map units. But ultimately this map unit and, right, this map unit or this centimorgan, it is same. Fine, it is same. And what else you need to remember here? That frequency of crossing over is equals to total number of recombinants divided by total number of progeny into 100. Fine, the frequency of crossing over is equals to total number of recombinants upon total number of progeny into 100. Like let's say total number of progeny is 1000, number of recombinants are 200. So tell me, it is the 20, frequency of crossing over is 20, frequency of crossing over is 20, yes or no? Yes or no? So that two genes are having a distance of 20 centimorgan. Fine, fine, they are having a distance of 20 centimorgan. Clear? So what you need to remember here, bache, always remember recombination frequency. This is very, very, very important. Recombination frequency is always less than or equals to, this is very important, less than or equals to 50%. This is a fact. Recombination frequency cannot be above 50%. It cannot be even 51%. Recombination frequency can be 50%. It can be less than 50%, but it will never ever be more than 50%. Like in a progeny, you can never say that recombinants are 600 and 400 are parental types. Never ever. It can be 500, 500. It can be 400 recombinants, 600 parental type. 300 recombinants, 700 parental type. But it will never ever be, recombination frequency can never ever be more than 50%. Clear, bache? It can never ever be more than 50%. Okay? Okay, Vidisha? So that is important. Okay, that is important. So see this, A and B, they are having the recombination frequency of 50%. So see, their distance is 15 centimorgan. So A and C, right, on the basis of this now, you can also, like let's say I am saying that the distance between A and B is, the recombination frequency in between A and B is 15%. I am saying in between B and C is 10%. And I am saying in between A and C is 25%. So, right, I can ask you to arrange them in a linear, arrange them on the chromosome. Now see, A and C, they have the recombination frequency of 25%, means they have a distance of 25 centimeter. A and B 15%, so 15 centimorgan. So see, here it will be 10 centimorgan. So that's how, okay, that's how you people can check. Done. That's how you all can check. So you need to solve question on the basis of it. Fine, you need to solve some question on the basis of it. Fine, bache. Okay. So next topic is of sex determination. So let's discuss this topic, right? It is our last topic, and then mutations and disorders pedigree analysis we will discuss in the next class. Mutations, disorder, pedigree analysis we will discuss in the next class. So now you know that for sex determination we have the chromosomes present, right? Isn't it? Isn't it? Chromosomes are present or not? Right? When it comes to the basis of sex determination, there are different things now, right? Chromosomal basis of sex determination is there na, chromosomal basis, like in the case of human beings, XX means female, XY means male. And even environmental basis of sex determination is also there. Environment also play the role in some of the animals, right? The temperature can, if there is an increase in temperature, female will form. If there is decrease in temperature, male will form. So environmental sex determination is also there. But as of now, in our syllabus, what are we discussing? Chromosomal basis of sex determination. Okay, chromosomal basis of sex determination. So here mainly your examples are important. Okay, mainly your examples are important. Like, let's see, most common type, XX and XY type of sex determination, right? So here this Y, it determines maleness. Okay, it determines maleness. Actually, bache, if I talk about the humans, fine, right? Female means XX, male means XY. This Y gene, this Y chromosome, actually this Y chromosome is having one gene, SRY gene, right? This Y chromosome is having gene, SRY gene, right? So, and even you talk about the testis determining factors which are present on these chromosomes. So in the case of males, this Y is very important. It determines the maleness. Okay, it is having the genes which will form the factors that will help in the formation of that male reproductive system. Okay, so this is very common in the case of humans. So you know that XX means females are XX, means females are homogametic. Females are going to form same type of gametes. They will have X or X chromosome. But in the case of males, they are XY, so they are heterogametic. Different types of gametes will be formed, right? So one will have X, another will have Y. So in the case of humans, whether the child will be a male or female, it depends upon male gametes. Females are not responsible for it, right? Earlier females were blamed when female feticide was too much. Females were blamed to have the girl child, right? That they are not able to give birth to the male. So it doesn't depend upon female gamete. Female gamete is same. Both females are going to form same kind of gametes. They will have X chromosome only. It is the male gamete, right, which will decide the sex of the baby. It is the male gamete which will decide the sex of the baby, that whether there will be a male child or the female child. Because if male gamete is X, female is already having X, so XX means daughter will be there. If male gamete is Y, female is having X only, so it will form the male child. So it depends upon the male. Clear, bache? Clear, bache? So males are homogametic, females are heterogametic. So you people can see the examples, isn't it? You people can see the examples as well. Yes or no? Tell me yes or no. So right now we are talking about the chromosomal basis of sex determination, also known as allosomic determination of sex. Fine. So this part clear? Yes, bache. This part clear? Sure? Okay. Sure. So if I'm talking about the mammals, like if I'm talking about the humans, so in the case of humans, 46 chromosomes are there. So can I write it? And yes, bache, see, 22 pairs in our case, they are autosomes. Your normal body chromosomes, they are known as autosomes, and that one pair, right, which is going to determine the sex of that individual, that is allosome. So sex chromosomes, for sex chromosomes, we use the word allosome, right? We use the word allosome. Are you getting it? Right? We use the word allosomes. Clear? So in the case of humans, 44 plus XX. 44 plus XX means 44, chromosome number 45, chromosome number 46, that's female. And when it is 44 plus XY, it's going to be male. Can I write it like this? In the case of humans, can I write it like this? So 44A, A is showing that autosomes, and XY or XX, it is the allosome. Okay, it is the allosome. Fine, it is the allosome. Now, in the case of insects, and especially, you know, your Drosophila. So Drosophila shows same human-like sex determination. That is very important MCQ, right? Drosophila, it shows human-like, right? It shows human-like sex determination. Now, you know, na, Drosophila is having total 8 chromosomes. When you talk about the Drosophila, so you know that Drosophila is having total 8 chromosomes, right? 4 pairs. So females, they are 6A plus XX. Males, 6A means 6 autosome chromosomes, and then XY. Is that clear? That's your Drosophila. Is that clear? A is showing autosomes here. Okay, A is showing autosomes here. Clear? Sure, bache? Then is XXXO type. So, bache, XX here again, that's female. XO is your male. So, in the male, 50% of the gametes, they will have X chromosome. This O is showing no gamete. O is showing no chromosome, right? O means nothing. So 50% will have your X chromosome and 50% will have your O. Clear? 50% of your chromosomes, they are having the O. So females have 2 X chromosomes, XX. Male possesses only 1 sex chromosome, that is X, right? Here males are having only 1 X chromosome, right? And females will have 2 X chromosomes. So such type of sex determination you will see in the case of cockroach, in the case of grasshopper, in the case of roundworms, right? In the case of roundworms. And moreover, bache, you know, this XX and XO type of sex determination is even seen in some plants, like your this Spherocarpus. This is extra. Spherocarpus. Okay, then comes ZZ or ZW. Here this ZZ is homogametic condition is there in the males. Usually we see na males are heterogametic, but here males are homogametic and this ZW is the female. So such type of sex determination in birds, in reptiles, in fish, in silkworm. Then comes the ZZ and ZW. ZZ again, here it is male. So in butterflies, in moth and pigeon and duck, such sex determination is there. So examples are important, right? Examples are important. And next is your haplodiplontic, right? Next is your haplodiplontic type of sex determination. And you are going to see it in the case of honeybee. Okay. Now you know honeybee is a social insect, right? Honeybee is a social insect. Okay, honeybee is having the queen, honeybee is having the worker bees, and honeybee is having the drone. So drone is the male member here, queen is the fertile women here, and they are sterile. Workers are what? They are sterile. And here, you know, what do you see? You see that these queen and worker, they are diploid, right? And male is haploid. Do you know why? Male is haploid here. This drone is haploid here. Do you know why? Because of parthenogenesis, right? Because of parthenogenesis. What is there? Parthenogenesis is there. What is the meaning of parthenogenesis, bache? The haploid gamete without fertilization, right? Haploid gamete, okay? Haploid gamete without fertilization, it is forming the adult, right? It is forming the adult. So without fertilization, when your gamete is forming the adult, this is the parthenogenesis. right? This is the parthenogenesis. So male member drawn is haploid and the female member is the diploid there. So this is haplodiplontic type of sex determination in honeybee. Females are having 32 chromosomes, males are having 16 chromosomes. Clear bache? Males are having 16 chromosomes. So see, mechanism of sex determination has always been a puzzle before the geneticists. Right? Before the geneticists, this mechanism of sex determination, it was a puzzle. So initial clue about the genetic or chromosomal mechanism, it can be traced back to some of the experiments carried out in insects. So see, what are they saying? The cytological observations made in number of insects led to the development of concept of chromosomal basis or genetic basis of the sex determination. So Henking in 1891 could trace a specific nuclear structure all through spermatogenesis in few insects, and it was also observed by him that 50% of the sperm received the structure after spermatogenesis, other 50% did not receive it, right? So Henking gave a name to this structure as the X body, but he couldn't explain its significance. So after further investigations by other scientists led to the conclusion that X body of Henking was in fact a X chromosome. Okay. So large number of insects, the mechanism of sex determination is bache XO type. Like 50% of the male gametes will have X chromosome, 50% will not contain it. Okay. So eggs are having additional X chromosome, right? In addition to the other chromosomes, right? That are the autosomes. I told you already. Clear? So some of the sperms, they bear the X chromosome, whereas some do not. So egg fertilized by sperm having an X chromosome become female, and those fertilized by sperm that do not have X chromosome, they form the males. Okay, so just have a look, bache. This is the example of Drosophila. So here they have even given you the example of the birds. So here you can see male is heterogametic and the male is homogametic and the female here is heterogametic. Okay, but in human and Drosophila, that's the scenario. Fine, bache. So grasshopper is an example of exotype. So examples are very important here. Fine, examples are very important here. So I already explained about the humans, about the Drosophila and the birds also. Okay, so see, mostly male heterogamity is there. And then see, it is explained in the case of humans. So you have to read it from NCERT. Okay. And the sex determination in honeybee is also explained to you. So see, in honeybee, it is based on the number of sets of chromosomes. So if offspring formed from the union of sperm and egg, that can be a female, that can be a worker. And if an unfertilized egg develops as a male, that is you're drawn by parthenogenesis, that male will have half the number of chromosomes than the female. So diploid female is having 32 chromosome and the male is having 16 chromosome. So it's not haplodiplontic, it's haplodiploid sex determination. Okay, instead of haplodiplontic that this term we use in the case of plants, no. So you can use haplodiploid. Yes, haplodiploid type of sex determination. So that's it. So it's not difficult, bache. This part is actually not difficult. Just focus on the example. So in the next class, we'll be starting with the mutation, the disorders, the pedigree analysis. Okay, mutation, disorder and pedigree analysis. So that's what we are going to study in the next class. And you know, bache, see, if you want to join our Avengers batch, because soon we will close the entries. I don't know when, maybe by the end of this month. Okay, and you should join it ASAP so that you can avoid the backlogs, right? At least be regular in the classes which are going on. See, if you talk about my syllabus, I am teaching human physiology as of now, right? By this month we will finish it and then we will start the class 12 syllabus. Fine. So you can see the link here. Just click on the link, add your number, your email ID, your details, and obviously you can be the part of that Avengers batch. Fine. And the PDF I will share in our official Telegram group, right? Join it. Fine, join the PDF there. Okay, and tomorrow mutations and the disorders we are going to study. So what you have to do, just give it a reading from NCERT. Fine, and solve the question on the basis of genetic mapping and all. You can take, like, let's say if A and B, they have the distance of 3 map units. Okay, B and C, they have the distance of 5 map units. They can even ask you to put that chromosome, put that genes in a linear order on that chromosome. So question like this can come. You should practice that. Okay, so you can go for any PYQ practice book. It can be MTG Fingertips, Disha publication, whatever it is, it's your wish, but start solving the questions. Okay, start solving the question. And guys, I need your few seconds. Just write down you like the session or not in the comment section. So stay tuned. Thank you so much. Bye bye.